Hello, everyone. I hope you are feeling wonderful. It's a pleasure for us share with you some aspects about our project called ShinyThor, two more high throughput omics-based roadmap on Shiny. Kind regards from Eduardo and Alexis, and we hope you enjoyed this presentation. First, what is ShinyThor? ShinyThor is an app implemented in R scripts and Shiny from RStudio, where the info is processed to generate examples of information and figures that will view in this presentation. We have an HTML implementation for the web version. This app that aims to support cancer researchers looking for a tool to evaluate expression profiles of genes, proteins, microRNAs, and metabolite presence in many cancer cell lines. Every estimations of these molecules in our app can be found in the units of measurement, such as relative units for microRNAs. Additionally, it is possible to run correlation comparisons to demonstrate whether the predicted inhibition radius of microRNA genes are validated in user-defined cell lines. Well, how is construct Shiny app? ShinyThor is implemented in a combination of R and HTML through Shiny app, okay? Where the front end supervises the visualization of the figures and tables built with CCLE information in the back end already loaded in the app. Okay. ShinyThor is built from information from the cancer cell line encyclopedia belonging to the DIPMAP, which aims to provide a dependency map for different genes in different cancer cell lines to develop new therapies, having an impact on precision oncological medicine, things. All the information is processed in R in order to generate a user-friendly interface, as well as in HTML for its web version, as we mentioned above. Well, what can we find? In the app, we can choose between two tabs, analyte expression and microRNA gene. These are two tabs present at this moment in the app, okay? In the first tab, analyte expression, it's possible to visualize the 15 results with the highest expression values or levels of genes, proteins, microRNAs, and abundance of metabolites in a bar plot figure, like the bar plot shows in this slide. Okay. For this, the user can filter by regions of interest like genes, proteins, metabolites, particular microRNAs, in a specific group cell lines, histological and pathological. Okay. Summary, we can choose in analyte profile gene expression, proteins, microRNAs, metabolite abundance, regions like TP53, CT protein, HSA, LED7B, or acetylcholine in the case of metabolites. Yeah. Cell lines can be bone, breast, pancreas, etc. Histologic groups like carcinoma, fibrosarcoma, neuroblastoma, and more. And pathological groups include benign neoplasia, metastasis, normal, or primary. Next, we can find a table can also be displayed in analytic data, okay? Analytic data, with the filtering of selected data to visualize all of the entries that match the choices in the group and region selection. Here, yeah. Summary. This is a summary of a query with information for all lines according to the query specification. And if we can check this table, note that the first results are the same showed in the bubble sorted from the highest to lowest level of the analytic with query specifications. Okay, check this. Check this. Are the same lines. Okay. Well, in the other tab, microRNA gene interaction, we can obtain a dot plot like this, displaying the correlation between genes and microRNA can be viewed according to their inhibitory activity. Okay, for example, yeah. Similarly, you can choose whether to filter by genes or microRNAs, then select the gene and microRNA of interest of cell lines, histological and pathological groups. Okay. Well, why is important ShinyThor? We have many reasons because it's important to study the cancer from tools like ShinyThor. We enumerate some of these. First, cancer is a multifactorial disease depending on intrinsic and extrinsic factors, such as acquired capacities to maintain proliferative signaling, resistance to cell death, oncogenic mutations, or exposure to carcinogenic agents, okay, etc. Cancer shows different behaviors according to the affected tissue, okay? According to the Rabier et al. In 2022. Therefore, the development of methodologies to diagnose and predict cancer takes a capital importance. With this high accuracy, it's important to update the treatment and survival standard. People. In consequence, ShinyThor seeks to provide information with the highest quality about genes, proteins, microRNA expression levels, and metabolic presence in many cell lines. Then, the app seeks to provide fast and simplified access to this information. Finally, in conclusion, okay, ShinyThor seeks to combine information from DeepMap in different cancer and genes, proteins, microRNAs, metabolites, okay, helps in identification of cancer biomarkers. They are relevant in cancer prediction and detection opportunity. ShinyThor seeks to assist in foster development by having updated and easy to view information. And finally, consolidate the app as a tool that facilitates browsing CCLE information and processing this information in a user-friendly interface, easy to interpret and mainly with a potential usability for cancer researchers interested in finding correlations between microRNAs, genes, as well as useful information of cell lines and the main modules of interest like gene expression, microRNAs, proteins, and metabolites in cell lines and pathological and histological groups. Well, this is all for our presentation. Thank you all. And remember, science is better together. This is our contact info if you want to know, to learn more about this, you can contact us at these emails. Okay. Thank you very much.